Hey everybody, just going to jump right into the notes on carbon and carbohydrates here. Okay, so the big idea is why is it carbon that's in living things? Functional groups, what kind of role do they play? How are carbohydrates and other molecules formed? And then how does the structure and function of carbs help life to exist? So we will run through those questions here. So what are organic molecules? All organic molecules have carbon. Okay, you guys probably know this already from regular bio or previous to that. They are found in all living things. Why carbon? Carbon's very abundant. Okay, we have a lot of carbon on the planet, and it's very versatile. This is really the main reason. With those 4 valence electrons, carbon can give away 4, it can take on 4, it can share. There's lots of different ways that carbon can interact with other elements, and so there's nearly endless combinations that you can make. So carbon takes on several different shapes, right? And why is that important? Well, remember, shape drives structure, structure drives function. So there's all these different functions that can come out of carbon-based molecules because of the shapes, etc. You have linear, you have ringed, you have branched, you have complex. If you're not sure what this thing is down here in the corner, in the bottom corner here, this is a protein. This is like a crystallized structure, or at least a computerized image of a crystallized structure of a protein. Okay. So Wohler and Miller end vitalism. Like vitalism used to be kind of before, too much science was known back in the early days. People just thought kind of life existed because of a life force, right? Like kind of like a Star Wars thing where there's just a force, and that's what why people were alive and why certain things were alive and certain things weren't. So Wohler and Miller and actually another guy, Urey, came up with this experiment down here, and so. This is actually the Miller-Urey experiment down here, and they were actually able to artificially generate organic molecules from this apparatus down here, and it kind of mimicked the conditions of early Earth. Okay, we're actually going to come back and talk about this diagram more when we get to our evolution stuff. But basically, these guys started trying to create some of the chemicals that we see in our bodies and in life. And started realizing that everything was kind of governed by these molecules, these tiny molecules and chemicals that are inside our body. Okay. So isomers, isomers are going to play a little bit of a role going forward. Different than isotope. Remember, isotope is different number of neutrons. Isomers is same molecular formula, different structure. Okay, so same exact number of elements are in each structure, but they're just arranged differently. Okay, so you see butane and isobutane here. Same number of carbons, same number of hydrogens, they're just arranged differently. So that's what an isomer is. And these isomers can lead to emergent properties. So when they rearrange, things can show up that weren't there previously, right? That's what an emergent property is. We talked about this with sodium chloride, okay, where you have sodium being the kind of grayish metal, and then chlorine being the yellow gas, and you put them together and you get the little powder that you put on your French fries. So that's what an emergent property is, and isomers can definitely lead to emergent properties. So a few different types here. You have structural, geometric, and a hard one to say, enantiomer. So structural, same formula, different arrangement, kind of the baseline there, what we just talked about. Geometric is cis versus trans. Let's see if the old pen's working today or not. Cis versus trans. So if you look at this diagram, here's the word cis and here's the word trans. So cis is going to be like adjacent to each other. So here we have these two hydrogens adjacent to each other, right next to each other. Trans is going to be opposite, right? Like transcontinental railroad, okay, like a cross. And so that's how you know if it's a cis or a trans orientation, depending on if the molecules off of that double bond are going to be on the same side or on a different side. And generally, geometric ones do have to do with the old double bond here. So you can kind of pick that out by looking at the double bond in the molecule. Okay. Enantiomer, same structure, but mirror images, kind of like a left-hand, right-hand thing. But a lot of times in biology, only one form is used. Both of them are not used, even though both may exist. What do you think would explain that. Pause the video, take a minute, see what you might think of. Okay, well, hopefully you thought about that for a quick second. Basically, it comes back to the structure function thing. It has to fit. A lot of times things fit like puzzle pieces. They fit like jigsaw puzzles. And they don't, if the left-hand version doesn't fit, you know, the right-hand version might. Like, you can't wear, at least comfortably, a left-handed glove on a right hand. Okay, so molecules are that way sometimes. They have to fit so specifically that they have to be faced a certain way or put together a certain way in order for it to work. Got to get rid of my pen to click forward. So here are some functional groups. I do not need you to memorize these. There are going to be some that come up more than others. You're going to see the amino group when we talk about proteins. You'll also see, probably not too much on ketones or aldehydes. You'll see the carboxyl group when we talk about proteins. Hydroxyls will come up a little bit as well. Methyl, just a little bit when we get to DNA. Phosphates show up a little bit, talk about ATP. Sulfates a little bit here and there. Sulfhydryl, not really. So just kind of showing you some of the functional groups that when you attach these to different molecules, they can definitely take on different shapes and then obviously different functions. And so you'll see a lot of these portions of molecules attached, these functional groups attached to molecules to kind of make them behave differently and give you certain functions. All right, getting into the macromolecules here. Macro, big, okay? These are really large molecules, okay? There are pretty common elements that make them up, right? And they usually start with CHO and CHONS and CHOMP, okay? So carbon, hydrogen, oxygen, you'll see nitrogen in there, you'll see a little phosphate in there, and you'll see some sulfur, okay? So some pretty typical ones are in there. Definitely are necessary for life, the macromolecules are. The four main ones are carbs, lipids, proteins, and nucleics. I'm not sure what happened to my picture right here. Looks like it got bumped over, nucleic. Sorry about that. Can you pick out which one is which of these four pictures? Pause the video, see if you can pick out which one of these four pictures is a carb, which one's a lipid, which one's a protein, which one's a nucleic. Okay, so pause the video real quick, see if you can do that. All right, so hopefully if we start at the top of the list, carbohydrate. Carbohydrate is going to be this one over here on the far right. Carbon, hydrogen, oxygen only. A lot of ring structures are typical of carbohydrates. We'll look at a lot more of those here in a second. Lipid. This one right here, the red and blue one, maybe that looks like a phospholipid. If you remember the phospholipids that make up cell membranes, there's lots of different types of lipids as well. This just happens to be a phospholipid. Protein. This crystallized structure down in here on the left that looks like a big ribbon that's all twisted, that's a protein. And probably one of the easier ones for you is the DNA, the nucleic acid, as the twisted double helix. I'm sure you guys are kind of used to seeing that. So how do these things become macromolecules? How do they get built? Okay, so some general terms are monomer and polymer. So a building block would be a monomer, and then a string of monomers is going to make a polymer, right? So if you think about the prefixes, mono is one and poly is many. Okay, so string of monomers, string of individual building blocks is going to make polymer, kind of like Legos. Individual Lego building blocks are your monomers, and then when you build something all up into whatever structure you're trying to build with Legos, then that's a polymer. Okay, lipids are the one that don't really have a true monomer. The other ones all do. Okay. And typically, the way these things are either built or broken down is by adding or removing water. And there's specific reactions for these. So dehydration synthesis, okay? So let's think about that real quick. Dehydration means to take water away from, right? To take water out of. So you dehydrate something, and synthesis is to build. So if you look at the words here on the screen, to make is synthesis, lose water is dehydration, okay? It's going to be endergonic because we're building, so we have to put energy in. Okay, and then building complexity is going to be anabolic, if you remember those words from regular bio as well, or even possibly chemistry. I'm not sure if they go over that or not. But anabolic is a reaction where you're building things up, right? And remember, anabolic, generally, all the people think of with anabolic steroids, right? And they're trying to build muscle and build things up into really big things. And so you can kind of relate anabolic to building from individual things into a bigger thing, okay? If you look at the diagram and you come over here, you'll notice that these two over here are just the monomers, okay? They are separate molecules right now, but they both have a hydroxide hanging off right here and right here. You have the hydroxide hanging off, okay? Well, remember, this is dehydration, right? So we're going to have to pull a water out of here. So if you look at the bottom section here, so dehydration synthesis, we're taking two things. This is one thing, this is another, but we're going to make it one thing down here. All becomes one molecule. So two individuals into one molecule that's joined. And so what happens is if you look from the red circle here down to here, we pull a water out. Okay, so here's your water getting pulled out. And so, sorry, it's kind of going off the bottom of the page there. So we basically take one whole side here. We have an OH, right? But we need another H. So we take that H and these two, and we make an H2O. And so that's where H2Os came from. And then what's left, the only thing that's left is that oxygen. So here's the oxygen left in the middle, and then it forms bonds between them, okay? So in this case, you're going from two monomers to one polymer by dehydrating. You're pulling water out. You're going to have a free water now, and then you're joining them through that oxygen, okay? So we're building a bigger molecule, and also we're storing energy, right? Because now there's a bond between the two, and the bond's going to store energy, which is why it's endergonic, okay? There's an input of energy. So if we look at the exact opposite, it's called hydrolysis, okay? Hydrolysis. Lysis means to split or to break, and then hydro is the water. So we're going to split water in order to break this thing down. Okay, let me grab my pen over here. So in this case, it's the exact same picture, but we're going to go in reverse. Okay, we're going to do the hydrolysis side. So now we have to split water, right? We're going to split the water, and this, whoop, let go. This bond is going to get broken as well. The bond right here with the oxygens, that's going to get broken as well. And when you break a bond, what happens to the energy? It gives off energy, okay? So hydrolysis is going to be exergonic, okay? Because we're breaking the bonds and we're giving off energy. And so when you take the bigger molecule here and you break the bonds, and we're going to stick the water right back up into the two monomers, the two building blocks, then it's going to go right back to being individual pieces. Okay, so hydrolysis is breaking down, and dehydration synthesis is building up. This would be an example of catabolic. Okay, so catabolic reactions that break things down, they reduce complexity, and they give off energy. Okay, so hopefully you remember a little bit about catabolic and anabolic. Moving into carbs. Okay. So carbohydrates, or, you know, the general term that we talk about is sugars, okay? They supply really quick energy, short-term storage, can't hold on to carbs for very long. Some of the sugars are actually for cellular structure, okay? There's a little bit for structure. So three big groups: monosaccharides, disaccharides, polysaccharides. Again, prefix: one sugar, two sugars, many sugars, okay? So only the ones that have many sugars, the polysaccharides, could be used for structural support, okay? The others just aren't big enough. We're going to go through each one of these, give you some examples. Glucose, right? Glucose is the main sugar of life, right? All living things kind of use glucose, the most essential sugar. C6H12O6, notice there's a nice 1 to 2 to 1 ratio there, and it's in a 6-carbon ring, okay? So the drawing down here is kind of all broken out, so you can see the 6-carbon ring. I do want to point something out to you in just a second. So if you look right here, C1, C2, C3, C4, C5, and C6. Those are not, those numbers on there are not the same as these, okay? They are not the same subscripts. They're not telling you how many are there. There are not 5 carbons there. That is carbon number 5. Okay, these are orientation. These are so we can talk about a specific carbon, and we know if something's happening at carbon 4, we can communicate about which carbon we're talking about. Okay, so they're just numerical positions that are assigned. Okay, all right. So a little more on glucose. Energy is stored in the carbon-hydrogen bonds of glucose. There's lots of carbon-hydrogen bonds, and that's where the energy is, right? The energy is always in the bonds. That's where the electrons are. And so the energy is usually stored in the bonds. And when glucose enters the cell, and those carbon and hydrogen bonds are ripped apart to release energy, right? That's what you do in cellular respiration, right? When you eat a meal and then respiration starts working, your bonds are just getting shredded in there, and every bonds that are breaking creating this energy. Okay. Galactose is a similar sugar to glucose. It's a stereoisomer of glucose, so just a different position on something, and I'll show you that difference here in a second, but also good for energy. 6 carbons in a ring structure. There's just one little difference. If you click back and forth between glucose and galactose, or if you actually look to the next slide, you can see where is that difference? Where is the difference between glucose and galactose? And it actually is here. If you notice the up and down here, right here at carbon 4 on both sides here, notice that the hydrogen is on the upside and the hydroxide is on the downside. And over here, the hydroxide is on the upside and the hydrogen is on the downside. But if you go around to each of the other parts, they're all the same. They're all identical. I know up here at carbon 6, here and here, they look different. They just happen to be written out different. This one just kind of listed the formula where this one drew it out. But if you count them up, it's all the same. Okay, so that's not really a difference. The only difference right here where they're flip-flopped. Okay, and actually, if you guys were, I know at South High, I don't know about the rest of you, but at South High and regular bio or even pre-AP bio, we build these molecules. Remember using model kits, maybe back to your freshman or sophomore year, and you built a glucose and a galactose, and you built one, your partner built the other, and then you flipped them, and then you joined them, and you made a disaccharide, and then you started joining them around the classroom, making one really big polysaccharides. You might remember doing that. You guys actually built these. Got to get rid of the pen. Doesn't let me click without the pen. So fructose is the third one. Okay, so fructose is really common sugar found in fruits because it's the sweetest. Okay, this is the sweetest tasting sugar. That's why fruit tastes so good. Little different arrangement. You can see it's got a different ring structure now because you do have the 6 carbons, but they're just arranged a little bit differently. You have another little tree over here. You have a structure here. Whoops. That did not let me do it. So here on carbon 6, you have this structure, and then you also have a whole other structure over here. So 1 and 2 are kind of combined. So now it makes more of a pentagon versus a hexagon kind of ring. So, so yeah, that's about it for fructose. Sweetest fruit, little different structure. Disaccharides now, 2 sugars. Okay, so 2 monos joined together. Okay, will always appear with 2 rings, and it's used to transport sugars. Now you can like join sugars together and ship them around your body, or a plant does the same thing. So how are they going to be combined again? What's the word for this? How do they get combined? Looks like they have it highlighted down here for you. Okay, so right here, right, we have those water molecules. They're going to be merging. The 2 H's and the O are going to be merging. We're going to pull water out. So this is a dehydration synthesis, right? Because we're building. Okay. So that is a glucose and a fructose that are going together. Okay. So we'll look at a few here. Sucrose. This is the sugar you guys have at home. Okay. This is your regular table sugar that you buy in the store. So this is sucrose. It's how plants store sugar. Chemical formula C12H22O11. Okay, and the structural formula is glucose and fructose. Okay, so it's a glucose, the essential sugar, along with fructose, the sweetest sugar, and that's why we like it so much. That's why it's in so many of our foods to the point where it's probably unhealthy. So if you look at the formula, why are these numbers what they are? 12, 22, and 11. If you think glucose and fructose are both 6-12-6, right? C6H12O6. So 6-12-6, if you have 2 of those, shouldn't it be 12, 24, and 12? If you just double it and you put them together? Well, it would be, except for you had to take out the water to get them to go together, right? So you pull out an H2O, so the numbers aren't quite the same, okay? So that's what it would look like right there. That's what a sucrose looks like, 1 glucose and 1 fructose. Maltose, okay? This is how animals transport their sugar, okay? Transport sugar using maltose. And this is, again, C12H22O11, and this is going to be 2 glucoses, right? So we're using a lot of glucose. All animals do, and so they are merging those 2 together in order to move them more efficiently, okay? So you pull out the water, and you get those 2 glucoses together, and you have some maltose. Oop, I forgot I had a little animation there. It's showing you where these are. Let me grab my pen again. It's showing you where these are merging. Okay, so the 1 here and the 4 here, they're talking about the carbons. Okay, this is carbon number 4 over here. Okay, right, carbon 4, and this is carbon 1, and we call this a carbon 1 to 4 linkage. It's the only place they'll ever join. Okay, because of the geometry of the molecules, it's always carbon 1 on one glucose and carbon 4 on the other is where they will join. That's where the hydroxide, the 2 hydroxides will go and pull out the water. That's just where that happens all the time. Okay, carbon 1 and carbon 4. Lactose, milk sugar. Okay, how sugar is transported to babies. So you have glucose and galactose for that. So mammals, when we have any mammal trying to get milk from their mom, they're using lactose, right? I'm sure you guys have heard of that. So you eat a disaccharide, you digest it into 2 monosaccharides. What must be added before this can occur? Pause the video. What must be added in order to take a disaccharide and digest it to 2 monosaccharides? What do you have to have? Pause it. Well, in order to do that, you have to have water, right? Because this is going to be a hydrolysis reaction. You got to be able to split water in order to make this happen. Okay, in order for it to go in reverse, you have to have water. So you have to have water to digest things, which is why you need to drink a good amount of water for yourself. That's one big reason why you need some water in your body. Polysaccharides, okay, the biggest ones, okay, many sugars. Okay, now we're talking about storing sugar for later and also for structure. Okay, polysaccharides are all polymers of glucose. It just depends on how they're joined together. Okay, so amylose is starch. This is going to be how plants store their sugar. Okay, amylose, starch, same thing, how plants store their sugar. So long straight chain. So let me show you. Here's glucose all going together. Okay, so all of those things are carbon 1 to 4. All dehydration synthesis to make that happen. You can see the oxygen in the middle of each of those molecules so that the hydroxide and the other H was pulled out for water. And then this one down here, this one down here is called amylopectin. It's the same thing as amylose, except for it's even more complex, where it's going to start breaking off and making different branches. More glucose is going to get added to that. Okay, so amylose plants, and then there is also an additional one called amylopectin. Okay. People throughout the world try to find parts of the plants to eat that are high in starch. Why is this? Okay, why do people want foods that are high in starch? Think about the foods that around the world people really need for energy. What is it? Well, it's things like corn and rice, because these are starches that are high in energy, right? It's because of all these carbon-hydrogen bonds. All the energy is in the bonds, and so they can get more bang for their buck as far as the energy is concerned if they're eating carbohydrates, especially people in parts of the world that maybe don't have a lot of food. You see a lot of things they make or that they try to make are out of like rice and corn and different dishes based around that, because they can get a lot of energy eating those carbohydrates because of all the carbon-hydrogen bonds and all the energy that's stored in there, right? And so what part of the plant do we usually eat to get starch? Well, a lot of the parts are underground even, right? Roots and tubers. So potatoes are underground, right? And potato, like a baked potato, is just a huge ball of starch, right? Just a huge ball of sugar, because that's where plants store their sugar, is in the part of the plant under the ground, like a potato, that's storing sugar for the rest of the plant that's above the ground. So a couple things to think about anyway, are just applications for how and why carbohydrates are important. All right, so glycogen is going to be a really important one because it's how animals store their sugar. Okay, so this gets stored in the liver and muscles, and it's really highly branched chains of glucose. So here's a picture for you, lots and lots of glucose strung together, and it's how organisms store their sugar. So you might have come in, or you were eating a meal where there was a lot of sugar, and so your body can't use it all at once, so it tries to store it in glycogen. And if you don't even use it within a normal amount of time, then it's going to get converted into fat. Okay. So cellulose, this is the first one where we're talking about structure. Okay. You guys probably have heard of cellulose. This is for structure in plants. Okay. Cellulose is in the plant cell walls. Okay, that's why they're very strong. That's why palm trees don't blow over when there's massive winds coming. They might bend, okay, but they usually don't break because cellulose is in those cell walls and it's so strong. Okay, so we'll show you a little bit more about cellulose here in a minute. But yeah, every plant diagram you ever see, you see that fat cell wall around the plant cell, and that's made of cellulose. And that's why plants could even grow vertically against gravity, because those cell walls stack on top of each other as they grow upward, and then that's what gives them the ability to actually grow up against gravity. Okay, all trees and plants have that. So a little bit more about cellulose. So the thing about cellulose, if you notice, is that if you follow the arrows as I click, look at the big structure, okay, that's up there by carbon-6, the big structure, but on the next one, it's on the bottom. And on the next one, it's on the top. And on the next one, it's on the bottom. And on the next one, it's on the top. So they're just getting flip-flopped every time with cellulose, right? So another way that the structure is varied with glucose, okay? But it's a long, straight glucose polymer chain. It's just they're flip-flopped every other one, okay? Here's a little diagram showing you how it kind of breaks down. So so here's your, your cell wall diagram, right? And you have this fat cell wall over here with, you break it down and you can see it's kind of stringy in here in the microscope picture. And then it gets back to kind of a CGI thing where showing you all of the fibers that come out of there. And then each one of these fibers comes out of there. And ultimately you get down to what the molecule is of cellulose. And it's all these ones, oh gosh, stop writing. It's all these ones where they are flip-flopped, right? So if you look, they're all flip-flopped here, okay? So all of those rows have the flip-flopped chains of cellulose, okay? Another feature about cellulose is we cannot digest cellulose, okay? So if you eat some celery, okay, it's in cellulose, out cellulose, okay? Your body's not gonna absorb any cellulose. We do not have the enzymes to digest it. So if you ever eat, cellulose, which, if you eat plants, if you eat vegetables, if you eat salads, you're gonna be eating cellulose, but our bodies can't use it. Some organisms have an ability to use it, and some organisms use other organisms to accomplish that, right? So like this one says some animals get help from bacteria or protists to break down the cellulose into energy-giving glucose units. We can use the glucose, we just can't break it down how it's organized. Our bodies don't recognize it, okay? So here you have termites, right? Termites are eating a lot of wood. They're eating a lot of cellulose, and they can't break it down themselves. But if you look inside their gut, okay, the stuff glowing in here, okay, the stuff all glowing in here, these are, they just tagged them in this kind of image. Those are all protists in there that can digest the cellulose. So it's a little symbiotic relationship between the termite and the protozoa and how to digest cellulose. So the protists are also getting some of that energy, but now the termite's getting the energy as well. So symbiotic relationship there in order to break down the cellulose. We can't break down cellulose, as I said. Some things can. Cows can, after kind of a long, lengthy process. So I don't know if you guys know what cud is, but cud is kind of how they constantly chew on grass, and it's basically like their bubble gum. They just make this big wad of grass and saliva and enzymes and stuff from their digestive tract, and they kind of swallow it, and then it comes back up a little bit, and they chew it again. And so they have a highly developed digestive tract. You can follow the lines and the arrows on there. You've probably heard cows have like many stomachs or whatever. Well, it's just kind of a long, convoluted tract in there. And so over a long period of time, they can break down cellulose. It just takes them a while. So that's why you see cows out there in the field munching on it and just chewing it for a while like gum. Because eventually their enzymes work on it. They just don't work immediately. They don't work, like you would think us breaking down food so quickly. It doesn't happen that fast for them, but they can get it done eventually. Another one is chitin. That is not chitin. That is chitin. So chitin is also a polysaccharide. And the main deal about chitin, if I go back here one, there's something really funky. There hasn't been any of our Sugars, any of the carbohydrates we've talked about up until this one, monosaccharide, disaccharide, polysaccharides, all of them that we've talked about so far are only made of 3 elements, carbon, hydrogen, and oxygen. This one's different because it has nitrogen, okay? It's the only carb that has nitrogen in it, and it's used for exoskeletons, as you can see over here in the picture, fungi cell walls, and actually dissolving stitches is made of this polysaccharide. So if you've ever had to get stitches, they stitch you up on the inside, and then the stitches just dissolve away into your body, and you don't actually have to get them removed. That's basically what this is, okay? So click off my pen here. So it shows you in this structure that you can see the nitrogen in the just off the ring structure there as well, and so a little difference for you there. Okay, so, and then the last one, carb we're going to talk about is peptidoglycan. Okay, so peptidoglycan you will find in bacterial cell walls. Okay, that's really the only thing we're going to say about that. But if you notice, there were 3 cell walls that showed up today. So peptidoglycan is found in bacterial cell walls. Which one was found in fungi cell walls? It was chitin. Chitin is found in fungi cell walls. And then which one was found in plant cell walls? And it was cellulose. Okay, so there are some structural ones, cellulose, chitin, and peptidoglycan, some of these polysaccharides that are good for structure. Okay, where a lot of the, the monos and the dis are mainly on the energy side, there are some structural carbohydrates that are used. Okay, so I think that's about it for carbon and carbohydrates. Here's a list of questions for you to practice with, and I'll get these posted as well, so you can just have the slides without me talking if you would like. But hopefully that helps you out with all of that. Okay. Take care.